All right. So the goal for today, and let me make sure that I have the right camera here. Okay. The goal for today is histone marks and chromatin 3D structure. Okay. So number one, we're going to talk about chromatin marks and their models, hidden Markov models, deep learning models, and then three-dimensional chromatin structure and how to infer it and how to predict it. So we talked a little bit about chromatin marks. Let's dive a little bit deeper. So as you know, we have 23 pairs of chromosomes. The X and Y are the funky pair. And then these are packaged up extremely densely in every one of our nuclei. So every one of our trillions of cells has 2 meters worth of DNA. If you put it end to end, as Professor Gifford said the other day, you would basically reach not just to Jupiter, but to the entire diameter of our solar system twice. So how are these packaged? They're packaged up into nucleosomes, and these nucleosomes have tails, and these tails have a lot of information. So basically, here's a nucleosome up close. You basically see that DNA is wrapped. You can see this lovely double helix. It wraps twice around every nucleosome. And the nucleosomes are basically made out of 4 components, H2A, H2B, H3, H4, each repeated twice. And there's these long amino acid tails that basically stick out. I don't know if you can see these little strings, but these are the tails of amino acids, basically amino acid tails of these histone proteins, okay? So this is packaging. This is extremely informational packaging, but it's also information. So this packaging contains a lot of information. So there's different histone variants that you use for different places. For example, histone H3.3 is used for transcribed regions. SENPA is used in centromeres. Histone H2A variants are used in place of DNA damage in the X chromosome in transcribed regions and so on and so forth. And each of those undergoes a large number of modifications. So these modifications are both serving to change the properties of the chromatin itself by condensing it or by loosening it up, but they also are read by a large number of chromatin regulators. So basically, these are organized in these domains, and these domains can be either compacted or loose. And then in the compacted domains, proteins have a hard time binding, and in the loose domains, they're much easier to bind. And you can see here that there's a very large number of different modifications that can be used to mark heterochromatic regions, polycomb repressed regions, promoter regions, transcribed regions, and so on and so forth. Okay. So here's just a very brief summary of some of the most abundant modifications. So we've been talking about H3K4 trimethylation. So this is histone H3. K means the lysine, and then the 4th amino acid. And then trimethylation basically means 3 methyl groups, H3K4Me3. H3K9 acetylation, for example, is associated with promoter region activation. H3K27 trimethylation is associated with polycomb repression. H3K27 acetylation is associated with enhancer regions. H3K36 trimethylation is associated with transcribed regions, and so on and so forth. Okay, so very large number of these modifications, and you can observe them using ChIP-seq that we talked about. And basically, by carrying out a chromatin immunoprecipitation experiment, you basically can see that promoter regions are marked with H3K4Me3. And when RNA polymerase, basically the protein that makes RNA out of DNA, is sitting, you can actually see it on the genome, and it sits in promoter regions and it starts transcribing. So there's many different types of signatures associated with different classes of elements. For example, you can find different classes of enhancers. You basically can find poised enhancers that basically show H3K27 trimethylation. This is a polycomb that basically keeps things repressed, but also H3K4 monomethylation, which is basically a mark of enhancers. When they become active, you basically see that H3K27 trimethylation goes away, and instead you have H3K27 acetylation coming up, and also this promoter-associated mark H3K4, sorry, this enhancer-associated mark H3K4 monomethylation. So H3K4 monomethylation basically marks enhancers, period. And then you switch from trimethylation to acetylation of H3K27. Raise your hands if you're with me on this one. Raise your hands if you're not with me. Any questions? Several people are not. So if you have a question, now's a good time to ask. So again, we're looking at the difference between having 3 methyl groups here, which basically marks polycomb, which is a repressive complex, versus having an acetylation mark there and therefore not having those guys, which basically marks active enhancers. So that switch is happening in these enhancer regions. Let me tell you one more thing. Who knows why there's a shape that looks like this? So why the shape? Why is there a dip in the middle? The reason there's a dip in the middle is because that's where the transcription factors are binding. That's where the nucleosomes are displaced. Where do you see these marks? You see these marks on nucleosomes. You don't see them on the DNA. And therefore, you see them to the right and to the left of the actual enhancer region. So the reason why they're peaking and dipping and peaking is because there's transcription factors binding here, and then the histone modification marks are flanking them, okay? So basically, if you look at this picture here, basically the transcription factors will be binding here. So what you will see is a peak, a dip, and a peak. When you see from far away, it looks like a single peak. When you look up close, you see the dip. And the dip is where the action is happening. At some point, we're writing a paper and we had an acronym for depletion in the intensity profile, also known as DIP. Anyway, so everybody comfortable with why do we see this change and why do we see this dip and why do we see these flanking regions with the nucleosomes being marked? Raise your hands if you're with me. Okay, raise your hands if you're not with me. Any questions? All right. Okay. So basically from far, far away, you basically see these huge peaks. And then when you look way, way close, every one of them basically looks like this, okay? So the question is, how do we learn this language of the chromatin? How do we basically take this chromatin immunoprecipitation experiments, not just for regulators, which is what we're used to, but now for chromatin marks and start interpreting them, okay? So how do we discover de novo the alphabet used for enhancers and for promoters and for repressed regions and for active regions and so on and so forth, okay? Basically, the alphabet looks like this. You basically can have, large-scale repression using either DNA methylation or using these kind of chromatic associated marks or these lamina-associated domains. So the lamina is basically the periphery of the nucleus. And repressed regions are usually stuck against that periphery, and that's called the lamina. And basically, lamina-associated domains is sort of one form of this massive repression. And then these are stably repressed, and then there's transient repression, and that's polycomb and H3K27 trimethylation. Okay. Within gene bodies, you basically have H3K36 trimethylation, H3K79 dimethylation, and these actually have different locations along the transcribed body. So for long genes, you see different transcription-associated marks early, middle, and late. Enhancers are basically marked with H3K4 monomethylation and H3K27 acetylation, whereas when they're repressed, you basically see this, repressive mark here. And then promoters similarly are marked with H3K4 trimethylation, and then just general acetylations wherever they might be. And then you can sort of switch from inactive to active, but also to poised. And then the poised ones have a combination of marks where basically you have some of these repressive marks, K27 trimethyl, and some of these active marks, K4 trimethyl. Raise your hands if you're with me. Yes, awesome, good. All right, so how do we learn the state of the chromatin, okay? So every one of these nucleosome bundles is 147 base pairs, and they're spaced by roughly a 50 base pair linker between one nucleosome and the next. So we'd like to sort of learn at some resolution, perhaps at some like 200 base pair resolution, the state of the chromatin. And one way to learn the state of the chromatin is to basically use combinations of these marks which together can basically give you a single annotation that basically says, oh, this is an enhancer region. I basically see H3K4 monomethylation. I don't see, for example, transcription, or I don't see, repressive marks like K27 trimethylation, K9 trimethylation, etc. And therefore, that marks enhancers. By contrast, this marks a repressed region. So here, I don't see any of the active marks. And instead, I see H3K27 trimethylation, polycomb, and H3K9 trimethylation, heterochromatin. Okay? And then the other thing to realize is the interplay between different types of repression. So DNA methylation marks most of the human genome. So if you look across the human genome, most of the places are repressed, except for small islands that are not methylated. And these islands are sometimes known as CPG islands. And the reason for that is that when you have a methylated CPG, a C-phosphate G, the methylated C turns to get deaminated and read as a T. So that basically means that when you methylate your CPGs, you actually lose your CPGs. And it's kind of interesting. So basically, you tend to have methylation most of the genome and very few CPGs in most of the genome. But in those CPG islands that are devoid of methylation, therefore they do not undergo this deamination process, you basically have less methylation. But they're having experiments that basically try to sort of remove methylation to see what happens. And it turns out that when you remove methylation, polycomb actually comes in and sort of represses these regions instead. So you see basically this interplay where when you don't have methylation, you actually have polycomb repression for several of these regions. Everybody with me here? Awesome. So we'd like to basically discover these states systematically. So the traditional approach for doing this is basically learning a probabilistic model that basically has a hidden state of the world and then observed state of the world. So the hidden state of the world is one of, say, 15 different chromatin states. And then the observed state of the world is what are the combinations of histone modification marks that I observe, okay? So the emitted symbol, Y, can be either open, closed, and then I choose what state am I in when I see open chromatin or closed chromatin, or it might actually be a vector of modifications. It might actually be a multidimensional vector. For example, I might have histone accessibility, chromatin accessibility. I might have histone modifications at every genomic locus. And every one of those nodes basically captures roughly the size of a nucleosome. Okay, so you could basically have 200 base pair chunks walking down the genome. So then the parameters are what is the probability of going to the next state from the current state. And these are the transition probabilities from going from one state to the other. And then the emission probabilities of what is the probability of emitting a vector of histone modifications given the current chromatin state that I'm in. Raise your hands if you have seen hidden Markov models before. Awesome. So this is kind of like a hidden Markov model, but it's a multivariate hidden Markov model. It basically emits not just one value, but a vector of values. Any questions? Any questions? All right. So one very popular implementation of such a hidden Markov model is Chrom-HMM. This was developed by Jason Ernst, who was a postdoc in my lab. And our idea was to divide the genome into 200 base pair windows and then have a hidden state for every 200 base pair window, which then models which histone marks are actually present in that window. Okay? So we basically just developed a vanilla HMM, which sought to model the epigenome and the complexity of these histone modification marks. Okay? And in contrast to a lot of what we've seen in this class, where we often build a deep learning model that tries to predict a class based on sort of previous annotations. This was an unsupervised model. So we just let that thing loose in the genome, and we basically said, hey, what did you find? And it came back and said, oh, I found 15 states. This is what they look like. And then we stared at the parameters that it had inferred for those states. And we also stared at the locations, thank you, where those states were occurring. And then we were able to name them enhancer, promoter, transcribed, repressed, heterochromatic, and so on and so forth. Okay? So basically it's unsupervised, and then the resulted states have to be then interpreted with independent data. And then the number of states was fixed and was a modeling decision. And frankly, we had this hunch as to sort of how many states should I have based on the number of marks that we have. Because the more marks you have, the more you're able to sort of distinguish different subtle states. If you only have one mark, there's really no point in having like 20 states. If you have 50 marks, then 2 states is probably underkill. So it was a right balance where, with more marks, we could actually learn more subclasses of elements in the genome. And we also developed a cool sort of nested initialization procedure that would basically first learn a larger model and then prune states progressively. And the reason for that is that we wanted to sort of enable the model to basically capture states from different initializations. And once it captured them, it knew that they were useful. Whereas if we sort of force the model to only have a fixed number of states, say 12, it may miss some important states, and we find some other states that are quite similar to each other. But by sort of first training for more states and then pruning down the number of states, that basically allowed us to sort of tune the complexity in a data-driven kind of fashion. Everybody with me so far? So this is basically what the model emits. It basically says, well, I have a set of emission parameters that basically tell me for every one of 15 different chromatin states, with what probability do I expect to see these marks? And it emits basically not just one mark, but a vector of marks. And these are the emission vectors. It basically says that in state 8, I mostly emit CTCF. And in state 4, I emit this combination of 5 marks. And in state 3, I emit K27 trimethylation, and this is state, and then these H3K4 monomethylation, dimethylation, trimethylation. So this is more of the poised promoter, and this is more of the polycomb repressed state. Everybody with me on the emission parameters? So basically this tells you which combination of histone modification marks do you expect to see in each of the states. It's a way of sort of summarizing the very high dimensional space of every possible combination of marks. And with, 8 marks, you have 2 to the 8 possibilities into a very small number, like 2 times 8 rather than 2 to the 8. Everybody with me? Then the transition parameters basically tell you what is the probability of going to the next state at position t+1, given that you're in a given state in position t. And that basically tells you that promoters tend to stay with each other, enhancers tend to stay with each other, and then these repressed regions tend to stay with each other. And mostly that you tend to stay in the same state for more than one 200 base pair interval. And that's just a feature of the data. It's just that, these histone modification marks tend to sort of have multiple nucleosomes marked at the time, rather than sort of switch from every nucleosome to the next. Everybody with me so far? So then this is what running ChromaHMM looks like using these, 6 marks that I showed you earlier, where you can basically annotate the, epigenome of this immune lymphoblastoid cell type based on promoters, transcribed, enhancers, repressed regions outside, and so on and so forth. And then these are the individual states that you transition through. And you can see this is a typical gene where you basically have the promoter, you have many enhancers within the gene body, and then sort of the next gene over, etc. And then you have some insulators, CTCF, for example, is this state 8 that sort of separate these from each other. Everybody with me? All right, so we developed this methodology, and it was collaborated with this large consortia to apply it systematically. So just a few years ago, we published this large consortium paper applying Chrome-HMM widely across 111 epigenomes, plus another 16 epigenomes from the roadmap Epigenomics project, from the ENCODE project, that basically capture, every major tissue in the body. Of course, this is bulk level. We are now working with many different consortia to basically try to infer this at higher resolution. So the International Human Epigenome Consortium has kind of taken the torch from the roadmap Epigenomics project and is working with ENCODE, with Blueprint, and with other places to sort of start marking the epigenome of many more tissues and many more cell types and many more individuals for each of those, and so on and so forth. And basically, this is what it looks like across many different epigenomes. So basically, this is a figure from that Roadmap Epigenomics Consortium paper where you basically see this PAX5 gene, which I showed you in the previous example. So you see here this is IMR90 lung fibroblasts, and there you can see PAX5 being repressed. But the question is, well, what does it look like across other epigenomes? So now that we have all of these marks summarized into one chromatin state track, we can now take 127 of these chromatin state tracks and put them on top of each other. And this is what that looks like, with basically PAX5 being repressed across basically almost every cell type we profiled, every tissue and cell type, except for just a handful where it appears to be escaping repression. And this is just a small number of immune cells. Everybody with me here? The other thing you notice is how stable promoters are. You can see that promoters, for example, are quite red, even though sometimes the genes turn on and off surrounding them, and then enhancers are extremely dynamic. So when you see a red column, it sort of spans the entire tree. And when you see orange, it very often is limited to just a small number of cell types. And that's because promoters are quite stable, whereas enhancers are much more dynamic across cell types. So can we predict chromatin state directly from sequence is the question. And using all of this methodology that you've seen so far, the answer is absolutely yes. So basically we can use these convolutional layers that we talked about at the beginning of this module to basically learn combinations of motifs, which then can sort of be put into higher and higher order constructs across upper level layers to basically predict chromatin state systematically. And there's been a number of efforts at predicting chromatin state using deep learning. So basically, these efforts include looking at the raw sequence of one cell type to basically predict simply open chromatin. These include looking at the raw sequence of one cell type to predict multiple chromatin states. This is multitask learning, where you're basically trying to predict an output vector that basically tells you what is the probability of each of 15 different chromatin states directly from the sequence. You can basically look at predicting accessibility across many cell types. You can basically look at, again, multitask learning, where you're basically trying to simultaneously learn the alphabet that predicts, 100 different cell types or 30 different cell types and their accessibility patterns. Why is that useful? The reason why it's useful to do this together rather than each cell type at a time is because those motifs, those convolutional filters that we talked about a couple of lectures ago, those convolutional filters are in fact shared across many of these tasks that you're learning. So if I were to learn each cell type separately, I would have to predict a set of motifs relevant to that cell type, and then go and learn a completely different set of motifs relevant for that other cell type, and a completely different set of motifs relevant for that other cell type. But in fact, the transacting regulators, the transcription factors that control all of this machinery are shared. They're just simply expressed or not expressed at various levels across the different cell types. So by basically doing this multitask learning of open chromatin across the many different cell types, I can sort of bring in that infrastructure that I've learned, these common convolutional filters, and then reuse them over and over again for each of the cell types. Raise your hands if you're with me on this one. Awesome, great. So these are just some settings, and not all of them have been explored in the literature yet. So I think this is a very cool area for sort of building projects for this semester. So basically, you know, this combination of, oh, how much can I pre-train the model by sort of using existing motifs? How much can I use multitask learning to look across cell types, across different chromatin states? How can I use additional information? For example, the transacting state of the regulators could be actually used as information to know whether these convolutional filters are useful or not. And I don't think this is out there in the literature yet. So basically your project could be, hey, let's have an additional you know, variable that controls whether each convolutional filter will be used by matching the transcription factor expression with the motif of that factor that I'm now discovering through these convolutions. You could basically look at the co-expression properties of transcription factors that basically tell you what is the cooperativity between these motifs. And those could actually be part of your upper levels or of your upper layers of your convolutional network to basically recognize which combinations of motifs are more likely to form at the higher levels, guided by the co-expression patterns of the regulators. You could use a recurrent neural network to basically learn how, as you progress across the genome, these neighboring regions depend on each other. How likely am I to transition from, you know, a polycomb state to an enhancer state, given the combination of motifs that are sitting underneath? You could be looking at using DNA accessibility information as an additional variable to predict the chromatin state of the next cell type, okay? So the reason why I'm telling you all this is to sort of get you guys to think creatively about what are all of the different modes with which you could combine everything you've learned so far to basically build predictive models. Raise your hands if you're with me here. Awesome. Any questions so far? Yes. That's so interesting. It's such a great question. Thank you so much for bringing it up. It's really a great question. So in this particular case, the HNM is working almost backwards from the convolutional neural networks. When we're talking about these convolutional neural networks, we have a sequence which is observed, and that's our X. And the Y that we're inferring is maybe, you know, pre-trained chromatin state annotation. So basically, if you were to build a convolutional neural network to basically learn enhancer regions, you would actually flip the X and the Y. The observations would actually be part of the input to the model, and then the X, which is the set of chromatin states, would be your Y, the prediction. In a hidden Markov model, things are flipped. And the reason for that is that it's a generative model. It's not a classification model. So the arrows are pointing from the model to generating the state of the world. And what the hidden Markov model tries to do is basically say, what hidden state is most likely to have generated the sequence that I observed? So it's a generative model that basically is trained to emit this sequence. And what you're doing is using Bayes' rule to infer the most likely hidden state that gave rise to the observations. Whereas with a convolutional neural network, what you would basically do is use the set of observations as input. And these observations could be the genome sequence, the DNA accessibility, maybe even the histone chromatin state. And then the output could be all kinds of cool things. For example, is this EQTL region? Is this a genome-wide association hit? Is this a functional non-coding variant? And so on and so forth. Great, great question. Thank you. I actually thought about it when I was describing this. I didn't want to confuse people, but you asked it at exactly the right time. Any other questions? Again, all these questions have been great, so don't be shy. If you're wondering, chances are other people are wondering too. Yeah, so again, another great question. So the question is, where do I get the gold standard reference of what is the hidden chromatin state of every region of the genome? Should I use the chromatin HMM output? And a related question is, if I use the chromatin HMM output, how can I go beyond it? Will I just be repeating what chromatin HMM already does? If you're trying to predict chromatin state from histone modification marks, and your training is chromatin HMM, effectively you're going to be reverse engineering chromatin HMM, which is fine. There's nothing wrong with that, because you can then use that for something else. You could use that for transfer learning. You could use that to refine it. You could use that to basically sort of force it to have a lower dimensional representation. You might be able to capture nonlinearities that sort of chromatin HMM does not capture, and so on and so forth. So that would be one way to train it. The other thing is you could actually, instead of having the same input, i.e. the histone modification marks, your input could be sequence. You could basically say, great, I'm going to build a multitask learning of predicting the 15 chromatin states from sequence directly in one cell type. So there, that's not what chromatin HMM is using. So then chromatin HMM is kind of cheating because it's looking at all of the histone modification marks to predict the chromatin states or to infer the chromatin state. And then you might be looking only at the sequence to predict the chromatin state, or you might be predicting whether a genetic variation will actually lead to a change in accessibility and so on and so forth. And that might be your prediction challenge. So these questions are great because they really sort of help all of you guys sort of think about what are the dimensions of the project. I can vary the input, I can vary the output, I can vary the type of model, CNN, RNN. I could sort of have additional variables that sort of guide the combinatorial nature or the higher order structure and so on and so forth. All right. So one such example, and I'm only going to show one because there's a plethora of these models and we saw some of them already, is this deep sea model. So basically, in this particular case, the goal was to learn TF binding, accessibility, and chromatin marks from the genomic data sequence through a series of layers to basically then predict what are allele-specific chromatin states based on the variation of the genome sequence, what are the effects of these on the chromatin state, and whether a genetic variant is in fact functional or not. And this is a hugely important problem because the vast majority of genetic variants associated with human disease are non-coding. So if you look at genome-wide association studies that basically look at common variants, and they've looked at, they've discovered 120,000 regions that are associated with human disease, 93% of those contain no protein alterations whatsoever. So they're likely to actually affect the gene regulatory circuitry of the genome. Many of them might be acting at the splicing level or at the microRNA binding level or at the RNA degradation level, but a big, big chunk of them actually act pre-transcriptionally. They're hugely enriched for DNA accessible sites that are associated with regulatory elements like promoters and enhancers, and they're hugely enriched for enhancers, enhancer chromatin states, and so on and so forth, okay? So that's why interpreting which of these genetic variants are meaningful is extremely useful, because the way that human genetics works is that we have inherited a small set of genetic variation from back when we were all living in Africa, okay? So humans lived in Africa for far, far longer than we've lived outside Africa. We only left Africa, you know, about, I don't know, a few tens of thousands of years ago. But we lived in Africa for hundreds of thousands of years since we diverged from chimps. So most of genetic variation, in fact, happened in Africa. And a tiny little part of that population, of that nice interbreeding, intermixing population, left and then populated the rest of the world. In Africa, that big chunk of variation still exists. Outside Africa, small chunk of variation is now distributed. So most of the genetic variants in the world are actually in Africa. A small minority of them are outside Africa, and they left. And then on those, just a small number of mutations also happen since we've been roaming the world for the last few tens of thousands of years. Everybody with me here? So now the problem is that when these genetic variants are associated with disease, we don't just have one genetic variant, we have 20 genetic variants. Basically, every chunk of the genome is inherited as a block. And the reason for that is that in that short time that the humans have been roaming the earth, there have only been so many recombination events. I only gave a small number of recombination events to my kids, usually on the order of 1 to 2 per chromosome. So that basically means that my kids have a huge chunk of my chromosome unchanged, and then a huge chunk of mom's chromosome, and then a huge chunk of my chromosome based on where those recombination events are. And over sort of population genetic time, you basically have these recombination hotspots where these recombination events are happening, and you need those to carry out crossovers and successful chromosomal disjunction during divisions. But then you basically get a whole chunk at a time. That basically means that when I look at a genetic association, even if only one of the genetic variants is in fact driving the association because it disrupted binding of this regulator here, I will have another 20 of them nearby that are along for the ride. And from genetics alone, I can't tell which of them is actually meaningful. And if I look at the chromatin landscape, this one might be active, I don't know, in liver, and this one might be active in heart. How do I know whether this genetic variant with this genetic locus is acting in heart or in liver? I need a computational model that allows me to predict which of these variants are in fact meaningful, which of these variants are having an impact. And the way to do that is to train a deep learning model, for example, DeepC in this particular case, that basically takes the DNA sequence as an input and looks at every genetic modification between them, and then directly predicts what will be the impact on TF binding, on epigenomic chromatin state, on DNA accessibility, and overall function. Raise your hands if you're with me on this one. Everybody understand the problem, why it's important? Genetic variation being co-inherited in big chunks, multiple genetic variants in each of these loci because of the history of the human population and those mutation accumulations, and then being able to sort of use all of these input data sets in order to predict the impact of the genetic variants. Any questions so far? I said a lot of things, so I expect some questions.
So I'm not asking 100% clear about what's the input to the what's form.
Yeah. So in this particular case, you basically have a lot of different features that you're inputting. You can input directly DNAs, TF, accessibility, histones. And then what you're trying to learn is for every window, what will be the impact of a genetic mutation. So for example, this was trained on 70% of the genome on all of these different profiles and excluding some particular chromosomes. And then what it was trying to, so basically the input is number one, the genetic sequence. And then the training is all of these different data sets. And then the output is what will be the impact of these genetic variations on the chromatin profile, and what will be the impact on sort of overall functionality of that genetic variant. Basically, is it likely to change, to impact the gene regulatory landscape? Yes. Yeah, it's such a great question. So will the model only recognize things that I train it and that I give it explicitly? There's many ways to think about that. One is that, yes, there might be some transcription factors that are missing from my set. Maybe I didn't observe a transcription factor at all. But there are other transcription factors that are co-binding with it. So maybe, even though the true impact is on a missing transcription factor that I've never actually given to my model, the model has learned that there's an impact here, which it observes through the other transcription factors that are coacting with it. So there might be shadows or ripples of the effect of that missing transcription factor on other transcription factors. So that's one way that the model can capture things that it did not see. The second one is looking across the genome, because it's looking at many, many hundreds of locations. It can basically learn motifs and grammars that are functional in many places, which it then can sort of use in additional locations. And the third one is multiple cell types. So basically, by sort of learning this language, quote unquote, of the gene regulatory circuitry in one cell type, another cell type, another cell type, you're suddenly getting glimpses of that missing data set that the model might be able to learn in this lower dimensional representation of your data. So basically, in these convolutional layers that it's learning, it's able to sort of capture information about missing data that you did not see because of all of this crosstalk across factors, across loci, and across cell types. Does that answer your question? Any other questions? Yeah.
So those genomic sequences that you use, are they germline sequences or something? And what do you need to do, like, there are sequence patterns that you like to make it as more robust?
To do a what? A per sequence?
Yeah, like a per sequence analysis.
Oh, paired. Yeah. So just let me start with the second question. So I can't do haploid analysis in the human genome, unfortunately. I only can do paired, because every single time I do a ChIP-seq experiment, I'm sequencing both alleles. So from the ChIP-seq experiment itself, I can look at the reads and sort of see, oh, there's a peak in this location in the genome. But that location contains a polymorphism. It contains a genetic variant. Let me see, where are the reads coming from in the peak? Are they coming from dad's allele, or are they coming from mom's allele in that one person? So even though I only have one cell, I have two genomes in that cell, mom and dad, and they're different. And their accessibility is very often different. If that genetic variant is actually functional, then, for example, the A allele might be leading to more accessibility than the T allele. Then when I look at the reads, I'll be like, oh, well, the genome is clearly diploid, but in that location, I have 40 T's and only one A. So that tells me that, oh, there's an allelic imbalance. So I'm in fact directly testing the allelic differences. So another project might be to go back to all of the ENCODE data and reanalyze all of the allelic reads to basically find places where the cis, like the genome differences between one allele and the other allele, are systematically leading to a difference. And then you can do that across these hundreds of data sets that I mentioned, because all of them are in fact diploid. And you can do that for, you know, many different ENCODE cell lines, many different ENCODE samples. There's NTEX more recently that's looking at primary sequence of individuals that are fully genome sequenced. So this might be another training set where you're looking specifically at the allelic differences between them. And then what was your first question?
Whether those were germline.
Yeah. So again, a fantastic question. So if you look at common genetic variation, we're talking about germline, and that's basically stuff that you're born with. But in addition to your germline, every single time that this, you know, initial zygote divides to basically make all of the cells in your body, it accumulates at some very, very small rate mutations. So some of those mutations might actually also impact, you know, gene regulatory function. Chances are they're not, because the vast majority of mutations are going to hit non-functional regions. So they might, and in any one cell type, even though across the entire genome, you know, we estimate that maybe, I don't know, 5 to 20% of the nucleotides might actually be impacting gene regulation or something if mutated. In any one cell type, that number is much, much smaller. You know, the 20% is like accumulating all of the different cell types. But in a one cell type, you know, there's only so many enhancers. So chances are that that one nucleotide that you got your mutation on is probably not going to make a big difference. And if it does make a big difference, chances are that cell is probably going to die if it makes a big difference. So anyway, there's one exception, which is cancer. In cancer, you have selection for mutations that are evading the immune system, that are evading all of the defenses that the host has, that are making it more, you know, likely to divide, to proliferate, etc. So in cancer, somatic mutations are extremely important. And that's where training these models, not just on germline variation, but also learning how to predict the impact of somatic variation has a huge impact for cancer. Or basically being able to say, aha, among all of these mutations that led to the selection of the virulence of the cancer, can I recognize which ones are more likely to be functional based on this predictive model? Who feels that they've learned stuff today? Yeah, awesome, great. So basically, DeepC indeed can predict differentially accessible regions just based on the SNP value. This is just some of the performance of how well it's predicting that impact. And you know, you could also build a logistic regression classifier using the DeepC output to identify regulatory variants, okay? All of this is treating now the chromatin landscape as a one-dimensional vector, where every location has some kind of information there. But in fact, more and more technologies have emerged for actually looking at how these locations are interacting with each other, because the genome, as it folds, has all of these higher order relationships. And in particular, one relationship that we care a lot about is how enhancers, which are these orange regions that are sort of far away from the gene, are in fact looping in 3 dimensions to basically bring all of these regulatory factors to the promoter, where polymerase will then sort of find a nice landing site and sort of sit, and then start transcribing the gene, okay? So this is, you know, known as looping, and this is bringing together enhancers with promoter regions. So one of the things that we want to predict is which enhancers are interacting with which promoter region, okay? And there's many technologies for capturing the 3-dimensional folding of the DNA. The most popular one nowadays is Hi-C. The way to think about the acronym for Hi-C is that it started with chromatin, conformation, capture, which was 3C. And then you had carbon copy, chromatin, conformation, capture, which was 5C. And then when Hi-C was developed, the inventors basically said, Erez Lieberman and Eric Ladender and Job Decker right here in Boston, basically said, oh, this is as many C's as you'll ever want. So we're gonna name it Hi-C. So basically what Hi-C does is it looks at this chromatin conformation capture. How? By basically cross-linking DNA, cutting it up, marking all of these cut segments with biotin, and then ligating the cut segments, and then selecting for the biotin markers, and specifically sequencing all those segments that had biotin label. That basically means that I'm sequencing junctions. Raise your hands if you get it. Awesome, great. So that basically means that I can figure out the 3-dimensional looping between blue and red regions by looking for chimeric reads in my biotin selected pairs of DNA segments. Everybody with me? So Hi-C has been applied massively and usually looks like this. You basically have diagonals that basically say that, ooh, things tend to interact, and off-diagonal items basically tell you about long-range interactions. And when you zoom way in, you basically see that the DNA is here, and then this particular segment here forms a long-range interaction there, and this one forms a long-range interaction there, and so on and so forth. Raise your hands if you're with me on how to read these plots. Awesome. So that also tells you that there are domains in the DNA, that this whole thing appears to be interacting more than anything around it. And this whole thing appears to be interacting more. And you see these like dots here? That basically means that there's a loop between this segment and that segment, and they interact a lot. Everybody with me? Awesome. So we can use this data to start modeling the three-dimensional contacts between enhancers, promoters, between domains of the chromatin known as topologically associated domains. And that's one technology, Hi-C, which is basically completely unbiased. Another technology is chromatin immunoprecipitation, followed by these ligations, which basically starts with all of the interactions that are happening surrounding a protein. For example, I could look at, I don't know, polymerase, and then ask what are all of the interactions that are happening when I pull down chromatin associated with polymerase. Okay, so ChIA-PET basically captures very similar information to Hi-C, but in a sort of through the lens of specific regulators. And this is just a zoom plot that basically tells you what are these interactions that are happening. Okay, so in the same way that you can sort of predict chromatin state from genome sequence, you could also basically ask, hey, can I predict 3D conformation from genome sequence? Okay, and there are many ways of doing this. There are many ways of sort of learning what are these interactions and then estimating these interactions and processing the data. So we're not going to cover this in any depth today, but basically after you've identified these interactions, you can now start asking, hey, can I train a deep learning model to predict enhancer promoter interactions? Okay, so Target Finder, developed by the Pollard lab, Katie Pollard, is basically using all of these different data types to basically directly predict Hi-C interactions. It's basically saying, great, I have all of this information, all of this one-dimensional information on many, many cell types, but I only have three-dimensional looping interactions on a small number of cell types. What if I could predict these looping interactions from these data? Raise your hands if you're with me on this one. Awesome, great. So what basically they did is feed all of that training data as input, basically looking at Hi-C and enhancers and express genes as input, and then looking at how can I predict this using the genome annotation, using CAGE, using ChIP-seq, using DNA-seq, using FAIR-seq, methylation, etc. And then holding out a set of enhancers and express genes that you can then ask, hey, am I able to use this input to now start predicting these missing data? Raise your hands if you're with me on this. Awesome. And the answer is quite remarkable. So basically, if you look at CTCF and RAD21, which is some of the transcription factors associated with these loops that are formed, you basically see that indeed they occur within interacting enhancers and also within non-interacting enhancers. And then when you run Target Finder, you can basically see that indeed it allows you to now start predicting where are all those sites occurring. So you can basically predict non-interacting as well as interacting loci with some probability, and then ask how well can I discover them. And then this is the performance in different cell types. And you can see that indeed there's a very strong ability to predict this information using all of these marks. Okay, so then you can basically build a promoter module, an enhancer module, and then sort of start asking, how much can I predict this based on sequence? How much can I predict this based on chromatin? And then build these fully connected layers to basically, combine all these features together, and then build these interactions to infer whether something is interacting or non-interacting using these combinations of features. Everybody with me? Awesome. So, and then, you can basically ask, what are the features that are driving these? What are the sequence features? What are the chromatin features? How well am I doing? And how well do I do with different enhancers? Who feels that they've learned stuff today? Awesome, great. See you guys on Thursday.